We're very, very thrilled to have Dr. Amburgey with us today to talk to us about genetics. And I forgot to introduce myself in the last session. That was horrible. So I'll do it here. My name is Nicola Worsfold. I am a mother of a 14-year-old boy with Duchenne. I also have an 11-year-old daughter. And I work at Jesse's Journey as the Director of Research and Advocacy. And we have Dr. Amburgey, as I mentioned, who is joining us to talk to us about genetics. She is a genetic counselor and a researcher at the Hospital for SickKids here in Toronto. Just a couple of housekeeping notes before we begin. You will be placed on mute, so your audio has been disabled, but please do ask questions. There is a chat box in your Zoom meeting where you can post your questions, post your comments, and we will monitor the chat and be sure to get to those questions during the question and answer period. And also a reminder that this session will be recorded. So just again, be mindful if in terms of sharing any personal information while the session is being recorded. But we also do encourage everyone to share the recording with your family members or friends who would have liked to have attended the meeting and weren't able to join us this weekend. So with that, I would love to pass it over to Dr. Amburgey. Thank you so much for joining us. We've been looking forward to this session.
Thanks, Nicola. Go ahead and put get my slides up. All right, is everyone able to see my slides?
Yes, we can see those. Thank you.
Perfect. Okay, so thanks, Nicola. My name is Kim Amburgey. So I am a genetic counselor at SickKids in Toronto, and I work in the neuromuscular clinic. So I'm very excited today to talk to you about genetics and how it applies to patients with Duchenne and Becker muscular dystrophy. Hopefully some of this is a review for some of you, and for some of you it may be new. So I'm happy to take questions at the end of the session. And just to give you a couple disclosures, so I have received a speaker honorarium from Novartis, and I'm a part-time employee at Deep Genomics. Today, I will be talking to you a bit about some basic genetic concepts. I'll be walking you through the genetic testing process for Duchenne and Becker muscular dystrophy. We'll review a bit about X-linked inheritance and touch on the options that are available for future pregnancies. Oops, sorry. So just to start with some basic genetic concepts, most individuals have 46 chromosomes. 22 of those pairs are the same among males and females. And you can see at the picture on the right that these are all of the pairs of chromosomes. The 23rd pair of chromosomes are the ones that are different, that make us male and female. So females have two X's and males have an X and a Y. And today we'll be focusing on that X chromosome, which is where the DMD gene is located. And we have two copies of every gene because we have two chromosomes, one that we get from mom and one that we get from dad. And as I said, Duchenne and Becker muscular dystrophies are caused by pathogenic or disease-causing changes in the DMD gene, which is located on the X chromosome. The DMD or dystrophin gene codes for a protein called dystrophin. And dystrophin connects the muscle fiber to the surrounding connective tissue and really acts as a shock absorber. So every time we're moving our muscles, you can cause a little bit of damage to that muscle. So that's what dystrophin does, is it protects the muscle. So in patients where this isn't working correctly, that's where we can see some damage and breakdown of the muscle. And this protein is really present in a lot of places in the body. So this can be in the skeletal and the heart muscles, as well as in the nervous system, including the brain and the nerves as well. And as I said, it's changes in that same gene that cause both Duchenne and Becker muscular dystrophy. And I like to think of dystrophin as a rope. So if we had a boat that's connected to an anchor by a rope. That rope is the dystrophin protein. And in patients with Duchenne muscular dystrophy, that rope isn't made really at all or incorrectly. So it really can't connect that boat to that anchor. And so the boat floats away. But in Becker muscular dystrophy, the rope is made. It may be a little bit shorter, but it can still function. And that's why patients with Becker muscular dystrophy tend to have a bit milder symptoms than those with Duchenne. Now, most patients with Duchenne and Becker muscular dystrophy go through the process of genetic testing. Some may go through a muscle biopsy as well, but really the gold standard for this diagnosis is genetic testing. And there are two main parts to testing for this condition. So the first part is the detection of deletions or duplications, where we're looking at the exons or the parts of the DNA that code for the protein. And we're looking for any deletions, which would be missing exons, or duplications, which would mean extra exons. And the majority of patients with Duchenne and Becker muscular dystrophy have deletions. So that's why we start there. If nothing is found in that deletion duplication analysis, we then move to sequencing of the gene. And our genes really only have 4 letters. There's an A, a T, a G, and a C. And what we do with sequencing is we look through all the letters of the gene, and we are basically looking for any spelling mistakes. Are there letters where there shouldn't be, or things like that? Some other important key terms that you may see on a genetic testing report are hemizygous, heterozygous, and pathogenic. So in boys, as we talked about with the sex chromosomes, they only have one X chromosome. So they only have one copy of the DMD gene. So any changes that we see there would only be present on that one X chromosome, which is where the term hemizygous comes from. In girls, they have 2 X chromosomes. So they have 2 copies of the DMD gene. And so the change on the DMD gene is typically on one of those copies, which is where the term heterozygous comes from. And then the last term is pathogenic, and that just means disease causing. So we're looking for changes that we know can affect the way that the protein works and would lead to symptoms of the condition. Now, let me walk you through what the results may look like when you get that report back. So from deletion and duplication analysis, we'll get to see that we're dealing with the DMD gene and where that deletion may occur or duplication. And just to give you an example, we'll be talking about a deletion of exon 43. And I also want to review one really important concept, which is the reading frame rule. So in general, this is something that's really important for deletions and duplications to know whether a mutation is in frame or out of frame. So in the DMD gene, there are 79 exons. So when the testing's done, they look through all 79 exons. They're looking for extra or missing exons. So in this example, if we have a deletion of exon 43, you can see in the picture there that exon 43 is missing, and you can't fit together those shapes in exon 42 and 44 don't fit together. So this is an example of an out of frame mutation, and this is typically associated with Duchenne. On the other hand, if we took out the next exon over, so exon 43 and 44, then we can see that 42 and 45 can now fit together. They are missing those 2 exons, but they're able to fit together and still make some of the protein. So this is considered an in-frame mutation and typically associated with Becker. Of course, there are always exceptions to the rule, but in general, most changes in the DMD gene do follow this rule. Now, if no deletions or duplications are found, the next step in the testing is sequencing. So they look through all the letters of the gene, and the report might look a little bit different. So I want to break down what a report might look like for those who have sequence changes. So again, we know that we're dealing with the DMD gene. And then there's 2 parts that we look at in the report. There's the part relating to the DNA and the part relating to the protein. So that middle box there is showing the change in the DNA, and then the last box is showing the change in the protein. And so, for example, in the DNA, if we are looking at what the change was in the DNA in this patient, we are looking at the exact position. So it's telling you that number there in the DNA box. This one here is at position 3,775. So that's the exact spot where there was a letter change. And in this case, it's a letter change from an A to a G. So that one letter was changed in the DNA sequence. Now, if we go to the protein, it's again telling us where we're dealing with in the protein. So it's at position 1,259. There's a change in the amino acids, which are the building blocks of our protein. So there's a change from an arginine, which is this ARG, to a cysteine, the CYS. So that one letter change in the DNA is enough to affect the amino acid in the protein. And in turn, if this were to be a pathogenic mutation, then we would suspect that it would affect the way that the protein structure or function, and therefore lead to symptoms in a patient. There are other types of genetic changes that we can detect. So for example, that's what we would call a missense mutation, but there are a few other types as well, and I'll just review these briefly for you. So if we were to think of our chromosomes like they were chapters in a book, one that we would get from mom and one that we would get from dad, our genes are like those sentences in the chapter. And then the mutations would be those misspellings. So it could be a change in a letter or a word. So for example, in the missense mutations, we're changing that letter or that word. So if the sentence was the car was red, we're now changing it to the car was hat or the car was RDD. Those sentences don't quite make as much sense. Another type of genetic change that we can see are nonsense mutations, and this is when the instructions stop too soon. So again, if we go back to that example, the car was red, this sentence would end at the car, and there would be no further information. We can also see smaller insertions or deletions, so this would be the addition of one letter or one word. And so again, if the sentence was the car was red, now we've got the car was red, or the car was red. Again, the sentence doesn't make quite as much sense. Deletions are the opposite, where we're missing a letter or word. So those are the four main types of changes that we can pick up through sequencing. Now you might be asking yourself, so what is the importance of having a genetic confirmation of the diagnosis? And there are several reasons to be thinking about this. The first one is to really confirm the diagnosis. And in some patients, we go straight to genetic testing, so you don't need to get a muscle biopsy, which is a somewhat invasive procedure. It also confirms the type of muscular dystrophy that a patient has. There are many forms, as I'm sure many of you are aware, of many different types of muscular dystrophies. And so this would really confirm that this is a dystrophinopathy or a Duchenne or Becker muscular dystrophy. Once a genetic change is identified, this also helps with testing of other family members so that we can identify women who are carriers in the family or other individuals who might be at risk for Duchenne or Becker muscular dystrophy. And then the third thing to be thinking about is consideration of treatment options. And there will be many talks throughout this conference in talking to you more about the specifics around the different therapies that are being developed. But as kind of a brief overview, there are some treatments that are being developed that really work on a specific genetic change. So some of these might be exon skipping. And this goes back to that reading frame rule that I was talking about earlier, where we want to put the reading frame back in frame. So if we went back to our example where we have the deletion of exon 43, what this type of treatment would do, it would then skip the next exon over exon 44, so that 42 and 45 can now fit back together. So this is an example of exon skipping, and it's really specific to that type of genetic change. There's also a therapy being developed called nonsense read-through. And so these are those nonsense mutations where the instructions stop too soon. And the therapy helps read through that stop sign basically and keep going so that the remainder of the protein can be made. And then I'm sure, as many of you have heard, for many different genetic conditions, CRISPR therapies are something on the horizon as well, which are those gene editing therapies. And there are also some treatments that are not based on specific genetic changes, So that an example of this would be gene therapy. So this is where we replace the dystrophin gene in a patient that's not working properly. And as many of you may know as well, this is a very large gene. So we're not able to put the entire gene into the viral vector to be introduced into the body, but mainly the most important parts are given back. And then there's also steroid alternative therapies that are being developed as well. So that's why it's really important to know the genetic basis of your disease, because it may affect the types of treatments that may be available to you, testing of other family members, and also confirmation of your diagnosis. So now that we've talked about the genetic testing and the importance of that and how to understand it a bit better, let's talk about the inheritance pattern. So as we've been talking about, the change in the DMD gene is on the X chromosome. And so this picture here is showing for those individuals where the mother of a boy with Duchenne or Becker muscular dystrophy is a carrier. So on one of her copies of the X chromosome, she has the change in the DMD gene, and that's represented by the orange X here. And she has one copy of a working copy of the DMD gene, which is shown by the blue X. And so let's split this up between boys and girls. So for every pregnancy, this is the same risk. So for boys, it's a 50-50 chance whether or not she passes on that non-working copy of the DMD gene. So for the boy that receives the working copy, he will not have the condition and not pass it on to his children. For the other 50%, she'll pass on the non-working copy of the X chromosome, and this boy will have Duchenne or Becker muscular dystrophy. So it's that 50-50 chance for her to pass that on to her sons. For daughters, it's the same thing, a 50-50 chance. For those daughters that receive the non-working copy, they'll be carriers just like her. And for the other 50%, they'll receive the working copy of the X chromosome and with the working copy of the DMD gene and not have the condition, not be a carrier, and not pass it on as well. So it's that 50-50 chance of passing it on to sons and to daughters as well to be carriers. And it's important to note that most female carriers do not have any symptoms, though some do have milder symptoms, and very rarely some females can have symptoms similar to boys with DMD. For those individuals who have had genetic testing, the moms of sons with the condition, it may have been found that the mother is not a carrier of the DMD change. And in this case, we consider the change to be de novo or a new mutation. However, the recurrence risk for these patients is still not zero. There's the chance that these mothers could have a change in some of their egg cells with the change in the DMD gene. And so we would quote a recurrence risk of around 10%. And this phenomenon is called germline mosaicism. So we can't rule out this possibility in females. We're not able to test that. So in families where the change is found to be new, we do quote a small recurrence risk in these families. Now let's talk about what the recurrence risks are for the boys with Duchenne and Becker muscular dystrophy. So again, in this picture, the X chromosome that has the change in the DMD gene is orange. And let's split it up again between boys and girls. So for all of the sons of a man with Duchenne or Becker muscular dystrophy, they will only pass on their Y chromosome. This doesn't have the DMD gene on it, so all of their sons will not have the condition. Versus the females, he only has that X chromosome with the change on it to pass on to his daughters. So all of his daughters will be carriers of Duchenne or Becker muscular dystrophy. So now that we've talked about the inheritance pattern and the genetic testing process, and we're able to confirm that in the family, we now know that there are options available prior to a pregnancy and during a pregnancy. So let's start with the option prior to a pregnancy. So there's a technology called pre-implantation genetic testing, or PGT. It can be used more broadly for chromosomal conditions, but also for what we call monogenic conditions like Duchenne and Becker muscular dystrophy, where we're only looking at one gene. And it combines two processes, so in vitro fertilization with some screening of embryos. So in vitro fertilization is where we combine the egg and the sperm in the lab. Those embryos are then screened for the change in the DMD gene, and only those embryos without the genetic change are implanted. There are several considerations to think about when choosing if this is the right technology for your family. One would be cost. So in Ontario, there is coverage through the provincial healthcare to cover one round of in vitro fertilization for a couple. But the screening of the embryos and some of the additional costs may not be covered. So there are some out-of-pocket costs, and that's something to consider when choosing this technology. In addition, the funding for IVF is also, there's typically a significant wait time. So that's something also to consider in the timing of considering this therapy or of this technology. And then when this technology is used, oftentimes multiple genetic samples are needed from family members in order to create the probes to be able to use this technology. So those are some things that are really important to be thinking about in deciding if this is right for your family. And then there are two options during a pregnancy. So one would be chorionic villi sampling, or CVS. This can be done both transcervically and transabdominally, and it's a way to test to see whether or not the baby has Duchenne or Becker muscular dystrophy. And so the way that this works is that a piece of the placenta is taken and the cells are analyzed to see if they have the change in the DMD gene that's been identified in the family. The procedure is done around 11 to 14 weeks of pregnancy, and the results come back in about 2 to 4 weeks. And it provides a yes or no answer whether or not the baby has Duchenne or Becker muscular dystrophy. And some families choose to make decisions about the pregnancy around this, or just to be aware so that they can be prepared. This is an invasive procedure, so there is a chance of pregnancy loss, and it's usually on the order of about 1%. The other process that is available is something called amniocentesis. Again, this is another invasive procedure, so there is a risk of pregnancy loss, about 0.25%. And the way that this technology works is that we take some of the fluid around the baby and test for the change in the DMD gene. This procedure is done around 15 to 20 weeks of pregnancy. The results come back in about 2 to 4 weeks, and again, it provides a yes or no answer. This procedure is available a bit more broadly than CVS, so more hospitals are able to offer this technology. So I know I've given you a lot to think about, and we've covered a lot of information, so I want to make sure that there's plenty of time to review your questions. So at this point, I'll open it up to questions.
Thank you so much, Dr. Amburgey. That was really, really great. And for some of us who, when we were diagnosed, we got a lot of this information. We kind of forget. So it's really good to have this again. I see we have one question coming in the chat box. It says, can you explain how exon skipping could work if multiple exons are deleted?
Yes. So let's go back. See if I can get back to those slides where we show the exons. So it's really dependent on which exons where the deletion occurs. But the idea is to find one exon on either side if there are multiple exons that are deleted to be able to put it back in frame. So even if a patient has a large deletion, if we're able to find a nearby exon that can be skipped to be able to put it back in frame, then that technology would work for that patient. But again, these are very mutation specific. So it's important when you're looking at the different clinical trials going on to make sure that your genetic change is something that's applicable to that technology.
Thank you for that. And please, I do encourage our participants to put your questions in the chat box. We're very fortunate to have Dr. Amburgey with us, so let's use her as best we can while she's here and ask away our questions. So we'll see if we get any more. I can certainly ask a couple of my own. And so first of all, I just wanted to ask, are all genetic reports reported the same? So regardless of where you live in the country, for example, the report that you get back, are they consistently reported in the same way? Are they reported in different ways? And sorry, you may or may not be able to answer that question, but I'm just curious for our families, when they're trying to sort of interpret that information, it might be useful to know.
Yeah, that's a great question. So there are many different labs across the country and outside of the country that do this testing. And the reports may all look a little bit different because every lab has their own way of doing things, but the technology is the same and the way that they report should be standardized. So that's why I kind of went through some of those important terms. They're things that you're often going to see. And so within the report, all of these elements should be there. So in general, they should convey the same information. It just may look a little bit different. So hopefully all of these, if you're able to kind of go back to this and pull out your report, you should be able to better figure out kind of what all of those different things mean.
Perfect. Thank you. We do have another question that came in. The question is, can you explain the basic concept of CRISPR? And we do have Dr. Cohen, who is going to be joining us later on this afternoon, who's going to be talking a lot about CRISPR. But Dr. Amburgey, can you perhaps provide us with some basic information or concept of how it works?
Certainly. I think I'm going to let Dr. Cohen go into that in detail. He's really the expert there. But the idea behind that is really that we can really target almost any mutation in the DMD gene and figure out a way to basically fix it. So this could be an exon skipping strategy could be used, or an intronic mutation, which is the part that is between the exons. That's more for processing of the gene, being able to target those, different sequence changes. So there's many different applications that CRISPR can be used for. And it's a very exciting technology and something that's on the horizon for patients with both Duchenne and Becker muscular dystrophy, and hopefully coming to clinical trial soon.
Excellent. Thank you. I do another question for you is, if we have individuals, young men perhaps, who've been living with Duchenne for quite some time, and they might not actually have access to their genetic report. And I'm not sure when they started testing genetically. Perhaps they didn't even have a genetic test. I don't know if that's even possible. I don't know if you can speak to that, but individuals who might not know their mutation and are adults living in the adult health care world, how could they go and get access to this information?
Another great question, and a really important question, and something, when I'm seeing patients in our clinic, I try and make it available to families because it oftentimes is very difficult to find in the medical records. Even if you reach out, if you know the hospital where you were followed previously, that where the genetic testing may have been done, it's often very difficult for medical records to find the genetic testing report. So if you're able to get in touch with either the doctor who ordered the test or the genetic clinic that you may have gone through, that's probably going to be your best bet in finding that report, because oftentimes it is very difficult to find it. It should be available somewhere, but oftentimes it is very difficult to find. So if you're able to go through genetics, that's usually the best way to do it.
Okay, that's great. And is it possible for them to request genetic testing, for example, like say they can't find a report? Is that something that someone could go back and say, I would like to get tested? And
Yep.
where would they, where would they go to get that?
Yep. So if testing was not done previously, if you're not able to find the results, there's many opportunities to try and be tested again or be tested for the first time if it wasn't done. So you'd want to go through, again, a genetics clinic to do that, to organize the testing. And they may also be able to help and if the testing was done previously to help you track down those results. But if it hasn't, there's many labs across the country and out of the country. There are programs that are available to access free testing as well, if it's something that's not covered by the provincial insurance or anything else. But it is a really important part of care for patients with both Duchenne and Becker muscular dystrophy to have access to that testing and be able to access the results so that they know what therapies might be available to them.
Oh, that's great. Thank you. We have another question that just came in the chat box. Can the same exon skipping technology be used for all exons? So we don't need a different trial for each exon we need to skip.
Also a good question. The technology and the idea behind it is the same, but there may be differences among safety for each of them. And so I know that's something that's under consideration for future trials. Are there ways that we can get more broad coverage or ways of testing so that we don't have to do a separate clinical trial for each one? But as of now, I think that's something they still are working on, and hopefully will be something that we can figure out in the future.
Absolutely. And those molecules aren't the same, right? Because each exon skipping therapy is targeted for a specific place on the gene. So in essence, they're a new molecule. They still have to go through the same testing process as every other new investigational molecule as well. Yeah. Another question, is the reading frame you showed on one of your slides reasonably accurate in terms of the shape differences? So I'm just trying to decipher, I'm not sure what they mean by the shape differences.
So I'm not quite sure about the question, but I think the idea behind it is just to give us a sense. Those aren't actually what those exons look like,
Yeah.
But it's the way that they fit together, and that we know which exons can kind of, if we were to put them together, would fit together. So it's really a representation of not exactly what they look like, but how they could potentially fit together. So we know which ones we could put back in frame together.
And are you able to share that slide again?
Sure.
Can we see them again to see the fit? But I think you're right. I think it's just a representation, right, of what they would look like and how they might fit together.
Yep. So are you able to see them?
Yep, we can
Okay.
see it.
Yep. So this isn't exactly what these exons look like, but the idea is that the ones that have similar shapes are the ones that we could fit back together to try and put the mutations back in frame.
Excellent. Any more last minute questions? I think we're a couple minutes away from starting our next session. Or no, no, my bad. I think we were supposed to end at 1:45. So I've taken away from your 5-minute break. My apologies. That was really, really fantastic. Thank you so much, Dr. Amburgey, for joining us today and for filling us in on all this information on genetics. It's always a lot for our families to digest all in one go, but every time we get this, it's like it keeps adding on to our knowledge, and really, really do appreciate that. So thank you. And our next session is going to be on participating in clinical trials. So I hope that I will see you all in 2 minutes. So thank you very much.